After chorionic villus sampling, how is the material processed for karyotyping? You should recognize when you see this word here, the kind of image you should be seeing in your mind is a bunch of chromosomes paired up like this, homologous chromosomes, and there should be 23 pairs. I'm not going to draw the entire thing, but that's what you should be thinking of. And these lines you're drawing are actually chromosomes, and the ones that are next to them are the homologous pairs. So if your dad gave you this one, that means your mom gave you this one. Homologous means they have the same genes in the same locations, but the alleles could be different forms. But we don't really care about that when we're doing a karyotype. The whole point of a karyotype is to tell us if we have a boy at the end for the last sex chromosomes, or a girl if they're both X chromosomes. Another thing to find out is if there's any kind of chromosomal abnormalities. So the 21st pair might have 3 of them, and that'll show up if non-disjunction has happened. So that'll mean Down syndrome is basically what we've figured out from looking at this karyotype. So chorionic villus sampling, if you don't remember that word, that's one way to get a actual sample of the DNA of the child that hasn't been born yet. And so the other way is through amniocentesis. Amniocentesis is taking samples from the amniotic fluid. Chorionic villus sampling is actually taking a bit of the tissue from a layer inside the placenta called the chorion. And either way, whichever process you use, this shouldn't freak you out if you don't know what this word is, but you can still figure this out. So what are we actually doing in order to create this karyotype? And hopefully you've already figured out by listening to the explanation here that we're looking for the homologous chromosomes that are paired. So why are the others wrong? DNA fingerprinting is not for karyotyping, but when you see an image or a diagram that looks like this, where you have gel electrophoresis that's producing little bands of DNA markers, that's going to be used for paternity tests or identifying criminals suspects in a crime, basically. Genes being photographed doesn't quite make sense. So what is a gene? A gene is a section of DNA, which if I took this gene out, it would just be a sequence of letters, A, T, C, C, whatever, a bunch of letters like this. You don't really want to be this is way too small to photograph anyways, so it doesn't quite make sense to photograph different genes. Alleles being compared is important, but it's not useful for helping us to process a karyotype. And remember, alleles are 2 different forms of a gene. So in my homologous chromosome here, if there's a big A, this has to be whichever trait is coded for by A, but it could be a different allele, so it could be little a. And so that's what alleles are. But in this case, how does the material need to be processed for karyotyping? D is the right answer. We want to take a picture of the homologous chromosomes and actually pair them together.